visible?
Yes, you are audible.
Are these lights? Yeah, and are these lights visible?
Yes, ma'am, visible, ma'am.
Okay. We'll wait for 2 more minutes and then I think we can start.
Okay, ma'am.
Hello, Ananta, you have raised your hand. Do you want to say something? Hello, Kshitij. Yes, tell me.
Hello.
Hello, yes, you are audible. Tell me. Hello.
Hello.
Yes,
Ma'am,
yes,
audible?
you are audible. Yeah, you are audible. Tell me. And
मैम मीटिंग पे काफी फेमस जो किस पर केस सिस्टम रहा है मैम
you are
ये क्लासेस
your
डेली मैम इसी टाइम
voice is breaking. Your voice
Hello?
is breaking. Yeah, your voice is breaking. What are you saying? Hello.
हेलो
हाँ, hello. Hello, Kshitij. Can you repeat whatever you said?
हेलो
Hello, yes, tell me, Kshitij, you are audible now. Hello, can you hear me? Hello, Janki sir, am I audible?
हेलो
Yes, you are audible, ma'am.
Okay. Hello, Kshitij, am I audible to you?
मैम मैं भी नेटवर्क इशू हियर मैम वॉइस शुड बी नॉट क्लियर
Okay, but I think network is fine from my side. Now I
Okay,
can
ma'am, I
hear
will check
you.
and
Tell
I
me.
will
Yeah, tell me now. You're
ma'am I
audible.
will be refresh okay
Huh? Yeah.
हेलो
Hello,
मैम
yes,
आई
tell
वांट
me.
जो क्लास चल रही है हम लोग की जो हेलो में नेटवर्क give some time I try to again join
Yeah, okay. You join again. Okay. Hello, Ranjit. Yes, you have raised your hand.
Hello,
You are
ma'am.
unmuted.
Am I audible?
Yes, you are audible. Am I
Ma'am,
audible to
will the
you?
recording of this session be provided to us?
Yes, sure. it
Okay,
will be
okay,
provided.
okay.
We will
Thank you,
send
ma'am.
you the link. Yeah,
Thank you so much.
yeah. The time. Hello, Janki sir. Are my slides in the presentation mode?
Yes, ma'am.
Okay. Okay. Hello, I welcome you all to the very first lecture of the genome editing class, and I hope you all are doing well. And we are going to have a really great time during this course. And since we don't know each other, I would like to begin with a brief introduction of about myself. So I am Sakshi Gupta. I have done my BSMS, it was a dual degree, bachelor's and master's combined in biological sciences from Iisc Bhopal and finished in 2018. Then I was a JRF at Iisc Bhopal itself, and finally I joined Iisc in 2019 as a PhD scholar. Right now, I am working with non-coding RNAs and I am doing my research in oral cancer. So let me walk you through the modules which we are going to cover in this short course. I am planning for 8 classes plus 1 maybe if it is needed. So 8 or 9 classes maximum. And I have planned to start from very basics because a lot of participants, so today around 400 participants have joined. So you all must be having diverse background. So I would like to start from very basics, and then from next classes we can increase the pace of the class. So today's class is going to be a little slow, and probably you must, if you are from a biology background, you must have been aware of these things. But it's a good idea to revise some basics before starting, before going into the detail. If you have any questions, if it is a small questions, you can raise your hand and I will try to answer if it needs some details, because we are so many here. You can write in the Telegram group, or I will try to answer them. And if it needs proper explanation, I will try to cover it in the next class. So so the syllabus and outline what I have designed for this course is as follows. We'll start with a brief introduction of genome editing, starting with general principles and classical experiments in biology. A brief history of genome editing, uh, some old methods like homing, endonucleases, ZFNs, TALENs. Then we will dive into the introduction of CRISPR-Cas, its principle and discovery, and its role in bacterial immunity. So this much portion I think will be covered in first 3 to 4 classes, and the rest of the 4 classes will be dedicated to the CRISPR-Cas only. So we'll discuss the mechanism of action, various Cas variants, the
Hello.
I think ma'am has.
Ma'am, you are not audible, ma'am.
Hello, Saki, ma'am. Hello.
Halo,
The meeting, I think.
Saki, ma'am.
ya, punten anu di
yeah,
dengar.
I think I lost the connection in between.
Okay, okay. Please share the screen.
Hello.
Yes, audible, ma'am.
Okay, okay. So I think I lost in between. Janki sir, can you tell me where I was, where I lost the connection?
Introduction, ma'am.
Yeah, just a minute. I'll share the screen again. So are these? So where did I lose the connection? Can you tell me? Hello.
Role in bacterial immunity. You just lost your voice.
Oh, okay. Okay. Thank you so much. Yeah. Okay. So as I was explaining, we will cover all these modules one by one, and this part will be done in the first 3 to 4 classes, and the next last 4 classes will be dedicated for the CRISPR-Cas. We will start with the mechanism of action. We'll discuss different variants. We'll design how to design your experiment, different bioinformatic tools to design the different requirements for the CRISPR experiment. Then we'll discuss how to generate the library. Then we'll discuss how the genome can be modified by CRISPR. Further, towards the end,
Ma'am, you're not audible.
Ma'am, you are not audible again.
Hello.
Yes, ma'am. Ma'am, you are again not audible.
No, wait, 1 minute.
Punten PPT-nya hilang.
Saki Gupta, ma'am. Saki, ma'am.
Aduh, ya teh kamarana atunya kalah Kak hilang.
Firman.
हाँ दूध तो खैर बढ़िया मिल जाते हैं।
Hello, Janki sir, am I audible?
It's audible, ma'am. Please rename itself.
Okay, okay. I am really
Yes, ma'am, you are audible. You are audible.
I'm really sorry, guys. I mean, I think there's some network issue. That's why. Mm. Okay, I think I was.
Saki, ma'am, please rename yourself, ma'am.
Hello. Yes, yes. So I think I was talking about the syllabus and outline of the course. And as I was mentioning, can you, somebody's mic is on. Can you please mute yourself? Yeah. So as I was saying, please mute yourself. I think many mics are on. Okay. So our class timing will remain same from 7:30 to 8:30 every Tuesday. And in case if there is any change, we will let you know beforehand. So to begin with, genome editing, the course Genome Editing Introduction to CRISPR-Cas. So genome editing is a branch of genetic engineering, and genetic engineering is defined as when we can alter genetic makeup of an organism, or we can construct all new genetic makeup of an organism. So we have fluorescent mice in the lab now. We have genome modified crops, and people talk about having CRISPR babies now. So let's see. So let's start with the course. So why we need to edit our genome? So the primary reasoning to study and understand various organisms. So it provides a great tool to, so genome editing provides a great tool to understand various organisms. You can delete the gene, you can mutate it, and then you can study it, what its role, what its phenotypic role as a treatment of diseases. So now gene editing is used as a hello.
You continue, ma'am.
Yeah, okay. Genome editing can be used as a treatment for various diseases. So further, CRISPR-Cas gives us a great opportunity to develop different various therapeutics. Yeah. Furthermore, in agriculture, we can if we can edit a genome, we can increase its yield so that we'll get more nutrients, and we can make them easy to grow. So this is another application of genome editing. And the last but not the least is to discover new scientific methods, scientific curiosity. So that's why also people discover new things, because they have scientific curiosity to know more about living beings. So this is example of a first see-through frog. So this paper was published in 2016, and here these scientists have made a first see-through frog. So in school we know that, right? We do a lot of dissection just to see what is there inside. So to avoid that, to avoid the to dissect many animals, these scientists have made these see-through frogs so that you can visualize, observe the organs inside by just looking at the frog from outside. So this kind of need is there. So this kind of experiments, this kind of experiments provides the opportunity to learn more about these scientific discoveries, and that's why we need to make genome editing more feasible. So first, to start the genome editing, first we should know what is a genome. So a genome is defined as a complete set of genetic information. Genome contains all the genetic instructions which is needed to make a body functional. It has all the set of instructions. You must have heard of other names like transcriptome, proteome. So transcriptome is defined as a whole, the amount of all messenger RNA molecules which is expressed by an organism, where the protein is defined as, proteome is defined as the total amount of proteins expressed by an organism. So unlike proteome and transcriptome, which varies from time to time and from tissues to tissue, genome is a very stable, it remains very stable throughout the life of an organism. It's like a blueprint of our life, and once, if you are creating any changes in the genome, it will permanently change the characteristic of that organism. So in 1958, Francis Crick explains uh, something called a central dogma, which says that a DNA, which is a genetic material, the genome for most of the organisms, transcribe into RNA, and then transcribe this process of formation of RNA from DNA is known as transcription, and RNA from RNA to protein is known as translation. So, and again, RNA can again be convert back to DNA by a process known as reverse transcription, and DNA can replicate itself throughout the life of the organism, which is and it's known as replication. So this whole scheme of the events is known as central dogma by Francis Crick. So talking about the genes, what exactly the genes are made of? A gene is defined as a segment of DNA that produces a functional product. So for many years, people thought a functional product should always be a protein, but this is not the case. A functional product can be a protein molecule or an RNA molecule. So now we have a lot of RNA molecules which does not code for a protein, but they perform certain functions, certain regulatory functions themselves without the need of coding for a protein. So the genes for these non-coding RNA molecules, together a gene is a segment of DNA that produces a functional product in form of a protein or an RNA. A gene is present inside the cell, inside the nucleus of the cell in the form of chromosomes. So these chromosomes are dense structures where the genes are present in the form of DNA, and these are very densely packed structure. And when we can understood it more here. So chromatin is organized in our nucleus in this way. So you can see here, this is a chromosome, and when you resolve it further, you can see there are a thread of structure. If you untangle the whole chromosome, you will get around a 2 meter long DNA. So this much long DNA is packed into the chromosome structure, and further, this DNA is wrapped around a class of proteins called histones. These histones, there are type of histone proteins like H2A, H2B, H3, and H4, and together 8 histone molecules. The DNA is wrapped around these 8 histone molecules, and this unit of DNA, which is wrapped around this one octamer, is known as nucleosome. So a nucleosome is defined as 8 histone molecules plus 146 base pair of DNA, which is wrapped around these histones. Here. So here again, you can see further resolution. You can see the histones and the DNA wrapped around it, which is this is also known as bead on a string model, because it looks like a bead on a string. And further you can see a double helix structure of the DNA. So at the time of expression, when RNA polymerase have to express the genes present in this DNA, this structure untangles itself and provide access to the transcriptional machinery. So the question, how many genes we have, this questions has been asked to us many times when we go for any interview or any other exam. They ask how many genes we have. So actually the correct answer is we don't know. We don't know exactly how many genes we have. So when you must have heard about Human Genome Project, which ran for around 12 to 13 years. And in the beginning of Human Genome Project, the scientists thought that there must be around 1 lakh, there should be around 1 lakh, more than 1 lakh genes present in the genome. And at that time, they were most focusing about the protein coding genes, because the non-coding RNA genes exist. This was not known at that time. Yes, but at the end, when the paper were published at the end of Human Genome Project, it was described that we have around 22,287 protein coding genes, and this data varies from databases to database. So here in this article, they have just explained how many genes we have, and then they compared that how many protein coding genes we have, how many long non-coding RNA genes we have, and this data differs. There is no proper consensus how much exactly, how many genes exactly we have. Like here this is 2000. This was published in 2018, and now when you check the number of long non-coding RNA genes, they are around more than 57,000. So it changes from time to time. So genome size, the size of the genome is related to the complexity of the organism. If organism is a very simple like bacteria, it utilizes it has a smaller genome size, so it utilizes its maximum of its genome size to make genes, because it has a very less genome size. But as we go further, as the complexity increases with higher organism, the gene density decreases. So gene density. So for example, here you can see a 65 kilobase pair region of DNA has been mapped, and in 65 kilobase pair region of for E. coli gene has a 57 genes, but in humans 65 kilobase pair region has only 2 genes. So genome size is roughly inversely proportional to the complexity of the organism. A brief introduction about chromosomes. You must have heard about ploidy. So ploidy is defined as a set of chromosomes. A haploid organism, if an organism is haploid, it means it has one set of chromosomes. And if a diploid organism has 2 set of chromosomes. Same goes on with triploid, tetraploid, etc. So here you can see a staining for chromosomes for human nucleus, and we can see here, since we are diploid, we have 23 pair of chromosomes, and out of 23 pairs, the meaning of 23 pair is we have one set of chromosome from our mother parent and the other set of chromosome from father parent. Out of 23 pairs, 22 pairs are autosomes, and while one pair XX and XY depends on the gender of the organism, are sex chromosomes. Some other terminology like homologous chromosomes, sister chromatids, it will be useful in our further classes, so I will just go through it. Homologous chromosome means if you got two chromosomes, one from if it's a diploid organism and it got one chromosome set from one parent, another from another parent, then these two chromosomes are homologous in nature because both of them have the location of same gene location. Both of them will have, for example, if this mother chromosome have the eye, the color for the eye gene here, the father chromosome will also have the eye color gene at the same location. So these two chromosomes are known as homologous in nature, and after replication they look like this because the whole chromosome has replicated itself, and then at that time these two arms are known as sister chromatids because they come from the same parent, and they, after replication, they have just double the amount of their DNA. And similarly, these two arms are known as non-sister chromatids. Uh, so how exactly the genes were discovered? So I think the first person who related the physical characteristics with with some factors which are present inside the cell was Mendel. And in 1866, at the time when nothing about DNA or RNA or any molecular biology was known, he connected that the physical characteristic of a plant, like height of the plant, the color of the flower, is linked. It's somehow linked to the factors which is present in the cell. And also he said these factors, which are later known as genes, are pure in nature. So like here, when he crossed one tall plant, one homozygous tall plant with a homozygous dwarf plant, he got all the tall plant. But when these two heterozygous tall plants were crossed, the dwarf plant popped up. So the dwarf trait was not lost, but it was there, but it will express only in its suitable condition. Yes, but unfortunately for many years, around 40 years, his work was not recognized. And in 1900s, three different scientists from different countries understood Mendel's work and presented in front of scientific community. And that time, everyone recognized the importance of the Mendel's work. After the rediscovery of Mendel, a lot of interesting discovery happened. Like in 1902-3, Boveri and Sutton independently proposed a theory known as chromosomal theory of inheritance. So the chromosomes were known, the genes were known, but genes are present on chromosomes were stated by chromosomal theory of inheritance. So which says that chromosome actually contain the genetic material which can be transmit from parent to the offspring or from one cell to another cell. These chromosomes can be replicated and passed along from one generation to another. The chromosomes are present inside the nucleus, and are present in pairs, in homologous pair in diploid organisms. And of course, if it is a diploid organism, one set of chromosome is inherited from the mother and the other chromosome is inherited from the father. Also, at the time of formation of haploid cell in the gametes, different type of chromosomes, non-homologous chromosomes, segregate independent of each other. So these statements are not just mere the statement, but are the result of many experiments. So also at the same time, Thomas Hunt Morgan, a very brilliant geneticist, and he showed that first time that a genetic trait was actually is linked to a particular chromosome. So for example, a white-eyed phenotype in Drosophila is linked to a particular chromosome. The gene for the white eye is present uh, on on a particular chromosome, and it cannot change. It is the position of a gene is fixed. So later, after around after 2 decades, another scientist, Frederick Griffith, performed an experiment which is famously known as Griffith's transformation principle. What he did was he took two strains of Pneumococcus bacteria. The two strains, one was known as R strain and another was known as S strain. R strain was a non-pathogenic strain, and if you inject it in mice does not die; it survives. But S strain was a pathogenic strain, and it kills mice straight away. So what he did was when he injected mice with R strain, mice was healthy. But when he injected mice with S strain, mice died. The next interesting thing he did was he killed this S strain. So at that time when the strain was heat killed, so now this strain was dead, so it did not cause any infection in the mice and mice survived. But another set of experiment, what he did was he mixed these two kind of cells. So he took R cells, R strain and the S strain and the heat killed S strain. So at the time when at, in this condition the mice died. So what was happening here? Why mice died when the S strain was killed and R does not cause any infection? So at that time, some of the DNA from heat killed S strain, which was present in the medium, was transferred to the R strain. So at this time, R strain became S strain and killed the mice. So whatever the genetic material is, we did not know that at this time that what is the genetic material, but whatever it is, it can be transferred from one cell to another cell. So this experiment is famously known as Griffith's transformation principle, and taught us that genetic material can be transformed from one cell to another cell. In another series of experiments which actually showed that DNA is a genetic material, because there was a lot of confusion. What is a genetic material? Is it DNA? Is it the protein? Is it the RNA? So every McLeod and McCarthy, they performed an experiment in which what they did was they took the cell lysate and then they added different so at this time again the different enzymes were discovered, like proteinase, which can degrade the protein, the RNAs which can degrade the RNA, and DNAs which can degrade the DNA. So they added these enzymes to different solutions of the lysate, and what they found out was, and they add R cells to it, and they found out at this time they got the S cells. So protein was definitely, because the protein was degraded in this case, so protein was definitely not mediating this transformation. Then they did the same thing with RNAs. When RNA was degraded, still our cells were transforming into S cells. And but when DNAs was added and DNA was degraded in the solution, and at that time there were no S cells were present. So this gives the first indication that DNA is the one which is actually genetic material, and which is actually gets transferred from one cell to another cell. Another 8 years later, very interesting experiment was performed by Hershey and Martha Chase, which confirmed that DNA is a genetic material. Till this time, also some scientists argued that no, protein can be the one which is a genetic material. So what they did was they took the bacteriophage, and they knew that at that time we knew that bacteriophage infects bacteria. So they radio labeled protein sulfur. So we knew that the sulfur was the part of the protein, and phosphate was the part of the DNA. So in one condition they radio labeled sulfur, and in another condition they radio labeled phosphate, and then they allowed this bacteriophage to infect the bacteria. Once this bacteriophage was inserted its genome into the bacteria, and then they wanted to see which of this element, sulfur or phosphorus, is seen in the progeny viruses. Interestingly, they did not see any radioactivity in the in the in the condition when sulfur was when the sulfur was radioactive, but they got radioactivity when phosphorus was radioactive. So again, only phosphorus is present in only in DNA. So only the DNA was the one which was getting transferred from one cell to another cell. So this experiment confirmed that DNA is the only one which is a genetic material and not the protein. At the same time, Rosalind Franklin and Maurice Wilkins, they took a very important picture, very important X-ray diffraction picture of DNA. This picture was responsible for the discovery of the double helix structure of the DNA, because in this picture it was clearly visible that DNA has a helical shape. Although it was clear that DNA has a helical shape, it was not clear whether DNA had two helix or two chains or three chains, but it was showed that there are more than one chain is present in the DNA. So here you can here they were also able to calculate the distance between the bases from this X-ray diffraction crystallograph. Also, the distance from one complete turn of helix was also found out from this crystallograph. So based on this image taken by Rosalind Franklin, two scientists, Watson and Crick, we all know them very well, solved the structure of DNA. And they said that DNA is a complementary. It is right-handed and it is a double helix molecule. It does not have three, but it has two helixes. So it has a polarity from 5 prime and to 3 prime and from both the direction. It is anti-parallel in nature. One strand goes from this direction, another strand goes from this direction, and it is complementary. So these two strands of DNA complement each other with and binds one base makes hydrogen bond to the other base and stabilize the structure in this manner. This lead to Nobel Prize to the Watson and Crick as well. So the structure of the DNA, I know many of you will know this, but just because this is just an introduction class, we'll just go through it. So what they found out, was there were two kind of bases were present: purines and pyrimidines. Purines were the double ring structural bases in the DNA, and the pyrimidines were the single ring structure. Uh, Two kind of purines, adenine and guanine, was present. It was 9-membered ring, and pyrimidines, three kind of pyrimidines. Two are present. Cytosine and thymine is present in DNA, and cytosine and uracil is present in RNA. Further, DNA, when sugar and base comes together, it is known as nucleoside, and the bond between sugar and base is known as glycosidic bond here, and beta glycosidic bond, where C1, first carbon of sugar, makes a glycosidic bond with ninth nitrogen of base. Say similarly with pyrimidines. So with the sugar and base together known as nucleosides, while when both the three things, which is DNA made of: sugar, base, and phosphate. When these three things comes together, it is known as a nucleotide. So based on which base is added here, we call it which nucleotide it is. If it is adenine or guanine, cytosine, thymine, based on that, it is known as by the name of that nucleotide. The bond between sugar and phosphate is a phosphodiester bond, while as I said, the bond between sugar and base is a glycosidic bond. So DNA is a complementary right-handed double helix molecule. Furthermore, another scientist, Chargaff, performed paper chromatography and found out what was the composition of these bases in the DNA. And when he calculated the ratio, it was very interesting, because the ratio of adenine to thymine was almost 1 in different organisms. So he took the DNA from different organisms, and he found that these ratio of adenine to thymine and guanine to cytosine is close to 1. It means the amount of adenine present in the cell is equal to the amount of thymine, and amount of cytosine is equal to amount of guanine. Furthermore, the amount of purines were also equal to amount of pyrimidines. This tells us that adenine always pairs with thymine with 2 hydrogen bonds, and cytosine always pair with guanine with 3 hydrogen bonds. One more experiment which was done by Messelson and Stahal. So till now we did not know how the DNA is getting replicated. So they performed an experiment in 1958. So what they did was they grow these bacterial cells in N15 medium, so that both of the strands of the DNA had the N15 nitrogen. And further, they allowed these cells to grow in normal N14 medium. So if DNA, since we did not know what how the replication of this DNA is happening, what they found out was one so here you can see, after centrifugation, since this isotope N15 was heavy, it was giving band here at a lower position. But after replication, when the cells were grown in 14 nitrogen medium, the band was a little up here, little up. Why? Because now the 14 nitrogen was incorporated in this double stranded DNA. And further, when they further allowed these cells to grow in 14 and nitrogen, what they found out was they found both the bands, 14-14 nitrogen bands and 15-14 nitrogen bands, which tells us that only one of these strands act as a parent for the for synthesis of a new strand. So a new strand will have only 14 and nitrogen. So here again, when these two strands will be separated, they will serve as a template for another strand, and the lighter strand will so lighter strand will act as a template, and both the strands will be lighter after a few generations. So that's why we will get only the light band. So this was only possible when DNA was following the semi-conservative, this kind of model. And that's how we came to know DNA replication. It follows a semi-conservative model of replication. Uh, Very brief introduction about DNA, the difference between DNA and RNA. So DNA is deoxyribonucleic acid, and RNA is made up of is known as ribonucleic acid. Why? Because of the presence of different kind of sugar. So here the two at the second position in the sugar, it's deoxy. So that's why it's deoxy. While the sugar in the RNA, it has a OH, so that's why it is known as ribonucleic acid. Also, DNA has two strands, while the RNA has only one strand. It's a single stranded molecule. The bases, as I have said earlier, the DNA uh, has four bases: adenine, guanine, cytosine, and thymine, while RNA does not have thymine but has uracil at the place of thymine. Another important thing we should always know is DNA undergoes, so after transcription, the RNA undergoes post-transcriptional regulation. So now we know that our DNA is divided in terms of, is explained in terms of exons and introns. Exons are the regions which actually code for the protein, but protein or RNA, the functional RNA, which I have explained earlier. And the introns are very long regions which gets removed after transcription. So here you will get the whole RNA transcript, and after the transcription, this primary RNA transcript will go under something called as splicing, which will remove all these introns, and we'll get only the exons together like this, and it will code for a protein. But but there is something called as alternative splicing as well, in which it's not needed that all the exons will come together only. One can come after three, or two can be missed, or three can be missed and two can come. And so in this way, it creates diversity at the RNA level and at the protein level. So now we know that different cell types can transcribe different kind of RNA and different kind of protein. So these proteins are known as isoforms of each other, and they can or cannot, or may not, can perform same or different functions. So this is another level of regulation which occurs after transcription. Furthermore, the post-translational modifications occur, where the protein undergo addition of different modifications like phosphorylation, methylation, etc. And these modifications also further affect the function of the protein. So with this, today we have just covered the basic experiments which were performed, which lead to the discovery of genome and genetic material. And it's important to appreciate these experiments, because these were the classical experiments which lead us to here. And this must be just a brush up for many of you, because you must have been aware of it, but it's important to revise these basics before going into the details. And in the next class, we will discuss more about the, we'll start with genome editing and the methods which have been used till now for genome editing. And some of the methods have received Nobel Prize for it. And slowly we'll move into the CRISPR-Cas. Thank you. So if you have any questions, I can you can raise your hand and I can unmute you. If you can also write the questions to me in Telegram group, I will try to answer them. And if it needs a more explanation, I will explain them in the next class.
Hello.
Yes, Akash, respond, sir.
Yes. Good.
Hello.
Hello. Can you hear me, ma'am? Good evening.
Yes, Akash.
Ma'am, it was really very fantastic presentation, and I learned a lot, and I hope so I will learn too much things again.
Thank you.
in
Thank you
next
so much.
classes.
Yeah.
Ma'am,
So
here my question
yeah.
is, ma'am, why? Ma'am, my question, why problem through means is there any reason
I
why
cannot hear you. I cannot hear anything. Can you repeat whatever you have said? Hello.
ma'am. Ma'am,
Yeah,
the
yeah.
thing is that hello.
Yeah, yeah.
Ma'am, why that frog name the true frog? What was the reason behind that? Simply that based on scientist desire.
I cannot hear anything. Sorry. Can you write to me your question? Are you there in Telegram group? Hello.
Yes,
You
ma'am.
are. Yeah, yeah.
Yes, ma'am,
You
I'm
are.
there.
Yeah. You can just write to me. Just search my name there, and you can write to me directly. I will surely answer your question. I cannot hear
Okay, ma'am.
whatever
Okay.
you're saying.
Thank
Yeah.
you.
Yeah. Hello. Sushmit. Hello, Sushmit. Yes, you are unmuted.
Ma'am, will be able to get this material?
Yes, I will upload all this and I will share you the link. I will
Okay,
upload
thank
to
you.
our, yeah. Priyam Vada. Hello. Hello. Yeah.
As you said, at the time of transition, some exons
Voice
just
breaking.
a minute. I cannot hear anything actually. Janki sir, can you hear her question? I cannot hear anything. Is it the problem at
No,
my end?
ma'am. No, ma'am. Voice breaking, ma'am.
Yeah. Voice is breaking too much, I think. Yeah.
Some questions posted in the Telegram group, ma'am. Please refer the questions also.
Yeah. I will do that. Also, I want to say I will share. Hello. Yeah, I will share a Google form with all of you. And please give your feedback. And I also need to know, I mean, how many of what is the background of the participants. Like some of you must be in BSc, just started BSc, so just passed the 12th standard, or some of you have cleared the MSc. So it's a very heterogeneous group of population. So that's why I kept this very easy. So just give your feedback. I will share your Google form with you and tell me if the pace of the presentation is it very slow, is it very fast, is it okay? You want me to skip these things and just move with the CRISPR-Cas, or I should cover the basics first and then go in further detail. I will share you the Google form and then you can just poll there. Yeah, okay. Hizba Gohar, can you, yeah, you are unmuted. Hello.
Hello.
Okay, hello. Yeah, I can see one question in the Telegram group. Somebody is asking, can you please explain the gene density and complexity part once more? Okay, we'll go through it one more time. Okay. Okay, here, right? Mohana. Yes. Yeah. So what happens, what I was saying is genome size is it's a rough correlation. It's not very exact, but it has been seen that the simpler is the organism is, it will try to use its entire genome to make only genes, right? So when you calculate gene density, which is genes per mega base pairs, so it will be more in the simpler organisms versus in the eukaryotes, in higher eukaryotes. In higher eukaryotes, like in humans, we have 3 into 10 raised to 9 base pairs. So this much big genome we have. So at that time, the gene density, when you will calculate, it will be much lesser. So here, like in, this is a span of 65 kilo base pair genome, and just a minute, this is hanging. Yeah. So in E. coli genome, across this 65 kilo base pair genome, 57 genes are present. But as the complexity of the organism is increasing, when you move to East Saccharomyces cerevisiae, in 65 kilo base pair region, only 31 genes are present. Further more complex organism like Drosophila, in 65 kilo base pair region, only 9 genes are present, and human, which is further more complex, in 65 kilo base pair region, only 2 genes are present. So as the complexity of the organism is increasing, the number of genes are decreasing, because a simple organism cannot waste its genome for with non-coding genes and all. It tried to make use of its maximum genome size. So that's why the gene density is higher in it. So I hope it's clear now. Yes, somebody is asking, can we leave the meeting if we don't have more doubt? Yes, you can. Uh, Somebody is asking about alternative splicing. Okay, I will explain exons and introns one more time. Okay, here. So our genes are divided in two forms. Some of the part is known as exons, some of the part is known as intron. Exon is the part which code for a protein, which will code for a polypeptide, for peptides, which will eventually will combine together and form a protein. But there are a lot of intervening sequences which are known as intron. So like here, exon 1, then there is the intron is known as intron 1, then exon 2, then intron 2, like that. And after this primary RNA transcript forms, this RNA will undergo a process known as splicing. What will happen in that uh, event? These introns will be chucked out of the RNA, and this yellow, green, purple, these exons will come together and it will form a transcript like this. So this is a final mRNA transcript, which has all the exons which has exons joined together. I will put another picture. This is actually this image is for explaining the alternative splicing. In normal splicing, what happens? Exon 1, 2, 3, 4, 5, 6 will come together and will form mRNA, which will form a protein. But doesn't happen like this. This is just a theoretical. Most of the genes undergoes something called as alternative splicing. What will happen in that condition is not all exons will come together. Some will come and some will also be removed with the introns only. So maybe in my heart cell and versus my lung cell, this is from the same gene, 1, 2, 4, 5, 6 exons will come together, but in my lung cell, 1, 3, 5, 6 exons will come together. Right? So this will give diversity at the level of RNA and at the level of proteins, because a different kind of protein will form from this mRNA and a different kind of protein will form from this mRNA. So these two proteins might perform different function in different kind of cells. So this I hope this is clear now. Otherwise, I will try to explain one more time. How do they get signaling like only 1, 3, 5 should combine? Garima is asking. I just want to confirm. Janki sir, am I audible?
It's audible, ma'am.
Okay, it's clear, right? I mean,
Yes, ma'am, clear.
Yeah. So okay. So 1, 3, 5. So what happens is at the end of 1 and at the starting of 3, there are specific sequences are present. So here 3 and 5 have to come together. So at 3 there will be some specific splicing sequence is present, and at 5 there is a specific splicing sequence is present, and there is a machinery called a splicing machinery which sits here and here, and brings these two exons together and remove this part, so that 3 and 5 comes together. These signals are present within the sequence itself, and it varies from cell type to cell type, tissues to tissue, organism to organism. Okay. Yes, introns don't code for proteins, so that's why they are removed. Yes, so they have to be removed because introns are actually, you can see, I mean, here you can see it's not very big, but and actually introns can be in the size of mega base pairs. Sometimes it's very in thousands, sometimes in millions. So they have to be removed for the proteins to be formed, because the protein is not that long. So exons are generally very short, like they can be they starting from 100 to 1000 base pair maybe, but introns are very long, sometimes in lakhs of bases are present in introns. But still cell can figure out where the introns are starting. And
Sorry.
hello, yeah,
Sorry.
somebody has to say anything. Yeah. How introns are removed, please tell clearly. Okay. I will put in the next presentation, I will explain the splicing. I will put all the proteins which are included in, which are responsible for splicing, and I think then it will be sure. But again, I will try. Like I have said here, if this intron has to be removed, some sequences, some are present here, specifically AG is present here, and in the beginning of the second exon, there is another a specific sequence, DNA sequence is present, such that these splicing machinery, these are proteins, okay? So these are spliceosomes. So these are RNA binding proteins. So they can bind to the RNA, the primary RNA transcript, and bring these two exons together and cleave these the bond between these between the intron and exon, so that these can form another bond. So I will, if you want me to explain the splicing in detail, I will keep a slide for splicing separately in the next class. Yes, is there any other question? Otherwise,
Okay, we will wind up the session, ma'am.
Okay.
Will someone raise a hand?
Yeah.
People already in the starting.
Okay, so okay, we'll just what is the time now? It's 44. The people who are raising hand, are, if you are in the group, you can write your questions in the group. I will answer every question. I will message you here, or I will, if it needs a explanation in detail, I will discuss in the next class. Okay, I think Angelo's. Okay, we'll just try. We'll take. Hello.
Hello, I want to ask you a question. I want to ask, how many courses are going to be in total in order to know how many weeks we need for this program?
How many what?
Courses.
Yeah.
In order to know how many weeks we needed for this program, I want to know if you know the exact number.
Totally 7 weeks remaining.
7 weeks.
I have planned for 8 classes. So including today's class, there will be 7 more classes, and depend on the response. If it needed, I will include 1 more class. So maximum 9 classes. So 9 weeks. Yeah.
Okay, thank you.
Yeah. Okay, thank you.
That's how you talk.
Okay. Chintaka, you can ask. Your mic is on.
Hello.
Yeah.
Thank you very much for the nice presentation. I'm from Sri Lanka, right?
Okay.
When you mentioned that our, let's say, genome is decoded, it means that they have discovered the nucleotide sequence of the chromosomal DNA, right?
Okay, when at what time you are saying? I mean, genome
No.
is
When we say that genome, human, the species genome is discovered or something.
Are you talking about Human Genome Project?
Okay. Or any other organism, doesn't matter.
At that time in Human Genome Project, the sequence was discovered. Before that, we did not know the sequence.
Right.
So it took around 12 to 13 years just to sequence the human genome, and after that, we have sequenced other organisms as well. Yeah.
Right. Then when we say that genome nucleotide sequence is discovered, it means that this sequence is a constant for a species, right?
Yeah.
Okay. So how can a person be different from another if this genome sequence is similar from one person to another?
I got it.
Okay. Thank you.
Yeah. So you know that, right? Our sequence and my sequence is around 98% similar. Same sequence. We and mice, but we are different organisms, right? So now we know that only the sequence doesn't matter, its regulation matters, right? I mean, there is like if I am talking about just as human, there is something called as epigenetics now. The regulation on my genome is different from the other person. I mean, if I am doing exercise every day, some other genes will express more in me than the other person who is not doing exercise. It's just an example. So that makes us very different. So even that small difference makes us all different. And the regulation is more important than the sequence itself, because if it is there, it should be expressed, right? Yeah.
Ma'am, if you can add this chatting box,
Here.
that would be better, because I'm not in that Telegram group. We can ask
Okay.
questions at the chat box. I don't see that chat box here. Thank you very much for your presentation, and thank you for the answer. Thank you.
Yeah, thank you, sir. Okay, sir. Hello, can you just can't you just join the Telegram group, Chintaka? Is that possible?
Yeah, it is possible. If you, I will answer the email that I received from you. So
Yeah.
if you can give me the directions, I will add myself to the Telegram group.
Yeah. So in the okay. So if you have the updated flyer, there also we have given the group link for Telegram group, and
I will see the email.
Yeah. Please see it and join, because I think it will be better there. I can just see anytime and then I can answer anytime.
Thank you. Thank you very much.
Yeah. Okay. Thank you. Okay. Nagul, you want to say something? Hello. Okay. Is there anyone has more questions? Nagul, you are unmuted. Nagul Kumar. Okay, I don't think.
Hello.
Veena.
Hello, ma'am.
Yes, ma'am. Yes, ma'am. Good evening, ma'am.
Yeah. Good
Thank you
evening.
very much for your very nice presentation. I just would like to know that whether the genome size remains static for every species, or it might change in time to come.
Yeah, it of course. Okay. Size.
Yeah.
I mean,
genome size.
Yeah. Size might take a long time, right? I mean, addition of new nucleotides will take a long time, and millions of years to be added to at least count them significant, right? Because we have 10⁹ base pair bases in one chromosome. So if you are added also, but it will take a long time, but sequence changes. So every time our DNA replicates, there are at least 3 nucleotides changes every time your DNA replicates. So with time, of course, you are not the same person you were born with. So your lot of sequence has been changed over your time, and when you pass your genes to your to the next generation, of course, these changes get transferred.
Thank you so much, ma'am. And I would like to join the Telegram group because I'm not in that. So I will see the instructions as you were explaining. So I will also try to join that. Thank you very much.
Yeah. Okay. Thank you.
Thank you.
Shrestha? Okay. Shrestha, do you have a question? Okay, maybe Syed Tariq.
Ma'am, excuse me, ma'am.
Okay, yeah, Shrestha, yes.
Where can I access the feedback from?
I will send. I have not sent it yet. I will send. I will send via mail and on Telegram group as well.
Okay, ma'am. Thank you.
Yeah. Okay. Syed, you have a question? Okay. Neeraj. Okay, I think then if there are no more questions, we can close the session for today. I will share the feedback form and please fill it. And let's see you all in the next class. Thank you.
Hello.
Yes, sir.
Can you stop the session, ma'am?
Yes. I just wanted to ask,
Ma'am, this is me, Neeraj.
Oh, yes, Neeraj. Yes, Neeraj, tell me.
Sorry, ma'am, I couldn't unmute myself at that time.
No problem. Yes, tell me.
Actually, I wanted to know about every time I read about structure of DNA, there is one term X-ray crystallography or refraction. Sir,
Okay,
if
yeah.
ma'am, if you could.
Okay. Okay, you want to understand the Rosalind Franklin picture? You message me on Telegram group. I will explain you there because I think most of the participants have left. So I will explain you there. Otherwise, I will keep in the next class again. Is that okay?
Okay, ma'am. Thank you.
Yeah. Okay. Thank you. Hello, Janki sir.
Yes, ma'am.
Yeah, was my voice fine after that? I mean, initially there were some network issue, but after that, was it okay?
Okay, ma'am. Good, ma'am.
Okay. Okay, then we can close the session. Thank you, sir.
Thank you, ma'am.